All right, it's important to note that all cells have a plasma membrane. Whether prokaryotic, eukaryotic, plant cell, animal cell, heart, nerve, does not matter. All cells have a plasma membrane. The plasma membrane is going to be found on the outer boundary of the cell and is going to be responsible for regulating movement of materials into and out of cells. Some cells have a cell wall that is found on the outside of the plasma membrane. So this will be external to, but it is important to note that the cell wall does not regulate what comes into or out of the cell. That is done specifically by the cell membrane. So it's common to all cells. It's the outer boundary of the cell. It's going to regulate the movement of substances into and out of the cell. And it's going to be composed of a phospholipid bilayer. It's ultimately going to separate the extracellular fluid from the cytoplasm, or the outside of the cell from the inside of the cell. And there are a lot of key characteristics of this plasma membrane. So there's a phospholipid bilayer that's going to make up the bulk of the membrane, but there are also going to be a great deal of proteins, cholesterol, carbohydrates. They're all going to be part of the membrane, and they give it these qualities of a fluid mosaic model. So we're going to start off though by looking at the phospholipid bilayer. Here is that phospholipid bilayer. You may remember this slide earlier from last chapter when we talked about phospholipids. Remember that there is a polar phosphate head and two nonpolar lipid tails. Does anybody remember what polar means? Polar. Polar means that this part of the plasma membrane, part of the phospholipid is going to be hydrophilic. Hydrophilic meaning water-loving. So this polar phosphate head is going to orient itself towards the extracellular fluid and the cytoplasm, both the outside of the cell and the inside of the cell. Because the extracellular fluid is primarily water, same with your cytoplasm, primarily water. These nonpolar lipid tails are hydrophobic. They don't want to be anywhere near the water. And so they are going to orient themselves sort of on the inside of this polar sandwich. So here again is just a little bit of a close-up. That extracellular fluid is going to contain water. There we go. That is going to contain water. The intracellular fluid, the cytoplasm, is going to contain water as well. The hydrophilic heads, the hydrophobic tails the hydrophilic heads are going to orient themselves on the outside, the hydrophobic tails on the inside. Oops, there we go. All right. Ooh, I'm going the wrong direction. Here we go. And then finally, proteins, cholesterol, and carbohydrates are also going to contribute to that plasma membrane. So we looked at the phospholipids. Now we're going to look a little bit at some of the other components of the plasma membrane. So here is our plasma membrane. We have our phospholipid layer, but we're also going to find proteins. Proteins are going to be embedded throughout the plasma membrane. Some are going to be channel proteins. They're going to allow for the passage of molecules back and forth. Same with the transport or the carrier proteins. We're also going to have some proteins that contribute to the identity of the cell. We're going to help identify what the cell is made up of, or what the cell's purpose is. Here are the three components of the plasma membrane. We've already talked about the lipid component, that phospholipid bilayer. But there's also the protein component. Protein molecules, they can be either peripheral or integral. Peripheral proteins are going to be found only on the cytoplasmic side, only on the inside of the plasma membrane. Integral proteins are going to be partially or wholly embedded transmembrane in the membrane, meaning that they're going to span that membrane. We're also going to find cholesterol. Remember, we've already sort of mentioned that cholesterol plays a very important part in the cell membrane. It's primarily going to be responsible for affecting the fluidity of the membrane. If we back up just a moment, you can see that we have, well, there's no cholesterol in this photo. So I'm going to move forward and see if I can find a photo. Here we go. So here's a close-up. We have our embedded proteins that are going to be spanning. We have our cholesterol, which is going to be found right here. It's going to prevent the close packing of the plasma membrane. It's going to prevent these lipid tails from getting too close to each other and keep it nice and fluid. Keeps the plasma membrane from becoming too tightly packed or too solid. So the next thing we're going to talk about is we care not just about the structure of the plasma membrane, but we care about the functions of the structure of the plasma membrane. Why is it structured the way it is? What is the point? So the plasma membrane maintains the structural integrity of the cell. It says it puts defined boundaries on the cell. It says this is the inside, that is the outside. We can go this far and no further. So it maintains the structural integrity of the cell. It is also, as we've already mentioned, semi-permeable or selectively permeable. This means that it regulates movement into and out of the cell. The plasma membrane not just regulates what goes into and out of the cell, but also plays a very important role in identification / recognition. There are several components of the plasma membrane, things like glycoproteins. Glycoproteins provide recognition. They sort of give what is known as a membrane fingerprint. They help identify the cell. Hey, I'm a skin cell. I belong to this human. I'm not a foreign invader. When we encounter situations where we don't recognize the membrane fingerprint, this can lead to an attack. Or if we don't recognize a membrane fingerprint, this can cause the body to cause the immune system to go onto the offense and start attacking that foreign invader. This is why it is so important to get a correct tissue match. This is why we care about blood type, but beyond blood type, we care about correct tissue matching. This is why organ transplants happen most frequently between relatives. There's a greater likelihood that their membrane fingerprint is going to be close enough that it won't cause the body to go into automatic attack mode and reject whatever organ is being transplanted. The plasma membrane also plays a very important role in cell-to-cell communication. There are going to be receptor proteins found on the surface of the cell that are actually going to receive messages from other cells and then respond. And so they're kind of like the walkie-talkie of the cell. They allow for cell-to-cell communication. Receptors are going to provide communication between cells. And then the plasma membrane may also have cell adhesion molecules that are going to stick together. This is what allows us to have tissues and organs. This is why you don't just melt or fall apart, because you're not just a collective of cells. You're a collective of cells that are stuck together, that are forming tissues that have a specific purpose. And so that plasma membrane is going to actually carry out a lot of different functions. So we're going to talk a little bit about that semi-permeable membrane, how we regulate movement into and out of the cell. And there are two types of movement across the plasma membrane. Sorry about that, I need to take a drink of water. There are two types of movement across the plasma membrane. The first is what's known as passive transport. Passive transport is movement across the membrane that doesn't require any energy. There are going to be a couple of examples of this. Simple diffusion, facilitated diffusion, osmosis. All of those are what are known as passive transport. No energy is involved in the movement of molecules across that plasma membrane. Active transport, on the other hand, is going to be different. It's going to require energy, movement across the plasma membrane that does require energy to get that going. And there are a couple of types: active transport, endocytosis, exocytosis. These all are going to require energy to move molecules across the plasma membrane. So we're going to start off by looking at this movement, starting with passive transport, specifically simple diffusion. Simple diffusion is just movement following a concentration gradient. We're going to be moving from areas of high concentration to low concentration, and the end result is going to be an equal distribution between the two areas. And so you can kind of think of this as a great example of this will be those air freshener misters. Have you ever seen those air freshener misters that, like, you walk by, it senses that someone's there, and it releases a puff of scent into the air? What you're going to find is in the area right around that mister, there's a very, very high concentration of scent. In the rest of the room, there's a very low concentration of scent. Simple diffusion doesn't require any energy, but it's going to say that molecules are going to move from an area of high concentration to an area of low concentration. They're going to move from the area around the mister, and they're going to slowly diffuse, slowly spread out across the entire room until the whole room is filled with sort of an equal distribution of the scent. So right by the mister, it'll start off being really, really strong, and in the far corner, I don't know, maybe it's smelly with a different smell. But given enough time, diffusion will cause that scent to spread evenly across the room. And the end result is we're going to reach equilibrium. Net movement is going to stop. Air molecules are still going to be bouncing back and forth and back and forth. Scent molecules will still be bouncing back and forth, but the net movement stops. We're not moving from areas of high concentration to low concentration. We're just all sort of bouncing around at equal concentrations. Much like if you were to pour cold milk into hot coffee. When you first pour it in there, you're going to see all of the milk is going to settle towards the bottom, but given a little bit of time, those molecules are going to move around and your entire coffee is going to be equally distributed with milk everywhere, or milk molecules are going to be evenly distributed throughout that coffee. Ultimately, over time, we're going to eliminate that concentration gradient. That's going to be the goal. There's no longer going to be an area of high concentration, area of low concentration. There's no longer going to be milk on the bottom and coffee on top. Given time, everything is going to be evenly distributed. And there are a lot of things that can actually affect this diffusion. Things like temperature, pressure, electrical currents, molecular size, all of these are going to affect the rate of diffusion, or how quickly molecules are going to move from an area of high concentration to an area of low concentration. When we are crossing the plasma membrane, there are few molecules that are able to move through simple diffusion, specifically and exclusively small and uncharged molecules. Anything too large or too charged is not going to get across the plasma membrane without some help. But small molecules, small uncharged molecules like carbon dioxide and oxygen are going to move freely across the plasma membrane. Why might this be a benefit? Why would we care about small and uncharged molecules moving freely across the plasma membrane? And why might we want to prevent large and charged molecules from moving across the plasma membrane? So take a moment and see if you can give me a good answer for that. So here is just a diagram of that. We're going to be starting off with an area of high concentration. We have a lipid-soluble substance. It's going to be moving through the bilayer, moving from areas of high concentration to areas of low concentration until we reach equal concentration on both sides of that plasma membrane. Along with passive transport, there's also going to be a process known as facilitated diffusion. Facilitated diffusion is going to be very simple. They're similar. We're going to be moving down the concentration gradient. We're going to be moving from areas of high concentration to low concentration. The difference being here that we're going to need a membrane protein to help us along. We're not going to be able to get through there on ourselves. So molecules that are too large or too charged are going to need help getting across that plasma membrane. They're going to be facilitated across by either carrier proteins or channel proteins. This is still diffusion. We're still moving from areas of high concentration to low concentration. We're still not requiring the use of energy. No energy is inputted. We're just helping out. It's kind of like taking a boat to float down the river. You can float down the river by yourself, or you can get in a boat and float down the river. It's just going to help move you along. This is what that looks like. Here we have a carrier protein. It's embedded in the plasma membrane. It's going to pick up cargo on one side of the plasma membrane and release it on the other. Again, we're not being selective. This is just movement from areas of high concentration to areas of low concentration. Osmosis is another type of passive transport. Osmosis is very similar to simple diffusion. The main difference here is that this is about the movement of water. Movement of water across a selectively permeable membrane from a region of higher water concentration to a region of lower water concentration. So we're still moving, but now we're concerned not with the moving of substances, but the moving of water. And the water molecules are going to move ultimately to dilute the solution. So we're just moving from areas of high water concentration to low water concentration. In our next video, we're going to talk a little bit about different types of solution and the movement of water.